All right, so today we are going deep into a world where computers are like molecular matchmakers.
Oh, that's a cool way to put it.
It's called computational drug design and virtual screening. Imagine a digital library, right? But it's packed with billions of potential drug compounds.
Each one, like a possible key to unlock a new treatment.
Exactly. And testing all those compounds in a lab. Forget about it.
Way too much time, way too expensive.
That's where the virtual comes in. Algorithms and simulations can sort through these molecular libraries so much faster.
It's like at warp speed almost.
It's like a digital treasure hunt for new medicines. So where do we even begin? The sources all mention molecular modeling as a key first step. How do we actually build these digital molecules?
So it all starts with the target, like a protein that's involved in a disease. Scientists can figure out its 3D structure, using techniques like X-ray crystallography. Then with some computational software, we can create a 3D model of a potential drug molecule too. This allows researchers to visualize how the drug might interact with its target at the molecular level. They can then predict whether it'll be effective or not.
So we're basically playing Tetris, but with molecules, trying to get that perfect fit.
Yeah, that's the idea. And the process of fitting a drug molecule into a target's binding site is called molecular docking. A good dock suggests the drug might be worth testing further.
And this molecular docking can really speed up the process, right? From what I've read, it sounds like researchers can test thousands, maybe even millions of compounds virtually.
Absolutely, yeah. And that's where virtual screening comes in. Picture a massive, super fast science fair, but it's all happening inside a computer. Algorithms sift through these huge databases of molecules to pinpoint those most likely to bind to specific targets.
It's incredible how technology is changing the pace of drug discovery. But the sources also mention some challenges with these in silico predictions. What are some of the limitations that we run into?
Well, the thing is, current docking programs, they're sophisticated, but they don't always perfectly reflect how things work in real-world biology.
Aha.
It's like simulating a golf swing on a computer. It might look perfect, but in the real world, you've got factors like wind, terrain, even the golfer's state of mind that can impact the shot.
Right. So just because a drug looks promising in a simulation doesn't mean it's going to be a hole-in-one when we try it out in a living thing. So what other hurdles do researchers face?
One of the big ones is accurately predicting a drug's ADME properties.
ADME.
ADME. Absorption, distribution, metabolism, excretion. It's like a drug's passport and travel itinerary through the body.
Ah, okay. So factors like how easily a drug gets absorbed into the bloodstream, how it moves to different organs, how quickly it breaks down, and how it's eliminated.
Exactly. All of those things can affect a drug's effectiveness and safety. Predicting these solely with computational models can be tricky.
And this is where AI starts to come in, right?
Mhm.
Particularly deep learning models.
You're right on the money. AI is starting to tackle some of these challenges, and one big development is AlphaFold.
AlphaFold. That rings a bell. It was all over the news for its ability to predict protein structures.
Exactly. AlphaFold can fold a protein into its correct 3D shape with amazing accuracy. This is huge because having accurate protein structures is absolutely essential for designing effective drugs.
So before AlphaFold, figuring out a protein structure was like solving a 3D puzzle blindfolded. And now we've got AI giving us a much clearer picture.
That's a great way to think about it, and having those clearer pictures means that we can potentially speed up drug development significantly.
So AI is becoming like a super-powered research assistant, providing insights that might have taken us years to uncover before. I'm curious, though, how else is AI being applied in drug design beyond just this protein structure prediction?
Oh, in so many ways. From optimizing the properties of potential drug candidates to predicting their toxicity and even figuring out how to repurpose existing drugs for new uses.
It's like having a multi-talented scientist who can work on many different aspects of drug development all at the same time.
Yeah, exactly.
And it's not just about speed, right? It's also about expanding the possibilities. Like, I read that AI can analyze huge amounts of biological data to identify potential drug targets that might have been totally overlooked in the past.
That's right. AI can look at genomic data, proteomic data, clinical data. It's just mountains of information that would be impossible for humans to sift through manually. It's like having a detective with superhuman abilities to uncover hidden clues and connect seemingly unrelated pieces of information.
It's really amazing how AI is transforming how we approach drug discovery, opening up totally new ways of understanding and manipulating these biological systems.
It's true, and as those AI models get more sophisticated and our data sets get more robust, we can expect even more groundbreaking discoveries in the future.
So we've talked about molecular docking and virtual screening, but what about the compounds we choose to screen? How do researchers decide which ones to test? Is it just random, or is there a strategy?
Oh, it's definitely not random. There are a few key approaches. One is called ligand-based virtual screening, where you use information about drugs that already work on the target to search for new molecules with similar properties.
So it's like saying, if this key works, let's find other keys that look similar and might also work.
Exactly. The idea is that molecules with similar structures probably bind to the same target and have similar effects. And that's where the idea of a pharmacophore comes in.
Pharmacophore, that's a mouthful.
Basically, it's a 3D map that highlights the essential features of a molecule that allow it to bind to its target. Think of it like a blueprint showing the keys, grooves, and ridges, the parts that allow it to fit into the lock.
Okay, so researchers create this pharmacophore based on drugs that we already know work, and then use that to search for other molecules with similar features, like using a template to find matching shapes in a giant library.
Yeah, precisely. This method can really narrow down the search quickly, but it does have limitations.
Like what?
Well, it relies on us already having knowledge about what works. So if we don't have enough information about drugs that already work on the target, it can be really difficult to create a pharmacophore that we can trust.
It's like trying to make a key when we don't even know what the lock looks like.
You got it. Also, this method might miss compounds that bind to the target in a completely different way than the ones we already know about. It's like assuming all keys need to look the same to open the same lock. Not always true.
So what are some of the alternatives?
Another approach is called structure-based virtual screening. In this one, you actually use the 3D structure of the target protein to identify places where drugs might bind. And then you screen for compounds that fit into those spots.
So instead of thinking about keys, we're looking directly at the lock.
Exactly. This is especially useful when we have a really high-resolution structure of the target protein, usually from things like X-ray crystallography or cryo-electron microscopy. It's like having a blueprint of the lock, allowing us to either design or find keys that fit perfectly.
And this is where molecular docking comes in, right? The thing we talked about earlier.
Yeah, exactly. We can use computational tools to dock tons of compounds into the target protein's binding site and look for the ones that have the strongest and most specific interactions.
Like trying to fit a key into a lock, testing different angles and rotations until we find the one that clicks into place.
And by looking at the results, we can prioritize the compounds that are most likely to block the protein from doing its job. The ones that can stop it from causing or contributing to disease.
This structure-based virtual screening sounds super precise. But are there downsides?
Well, it relies on having a really good 3D structure of the target protein. If the structure isn't accurate or is missing pieces, it can really mess up the results.
It's like trying to design a key from a blurry picture of the lock. It might not work the way we want. Anything else?
Another thing is protein flexibility. Proteins in the real world aren't static. They move and change their shape.
Oh, okay.
And those changes can impact how they interact with drug molecules.
So it's like the lock isn't perfectly rigid. It's got a little wiggle room, and that can change how well the key fits.
Exactly. And while there are some advanced algorithms that try to account for that flexibility, it's still a big challenge.
Now, you mentioned that there's a third approach to selecting these compounds.
Right. It's called pharmacophore-based virtual screening. And it's sort of a hybrid. Instead of getting the pharmacophore from drugs that already work, we get it from the 3D structure of the target protein itself.
So it's like we're using the blueprint of the lock itself to figure out what the key needs to look like.
Exactly. This lets us screen for compounds that match the pharmacophore, kind of like using the shape of the keyhole to look through a giant catalog of keys and find the ones that have the right shape.
It sounds like these different virtual screening approaches are a really powerful toolkit for drug discovery.
Definitely. And each one has its strengths and weaknesses, so researchers often use a combination of them.
To up their chances of success.
Yeah, exactly. Choosing the best approach depends on things like what data we have, what the target protein is like, and what the goals of the research are.
It's fascinating how these computational methods aren't just speeding up the process, but also helping us better understand how biological systems work. It's opening up so many new possibilities.
It really is revolutionizing drug discovery. And it's only just the beginning. I think as these technologies continue to improve, we're going to see some even more groundbreaking discoveries down the line.
Yeah, it's pretty incredible how far we've come.
Yeah.
But, even with these amazing tools, finding a molecule that binds well is just the first hurdle.
Oh, right. It's easy to forget that a drug might fit perfectly in the computer, but then have to survive a whole journey through the human body.
That's exactly it. And that's where ADME comes in: absorption, distribution, metabolism, and excretion. A drug's trip through the body can get pretty wild.
So it's not enough to find the right key for the lock. We also have to make sure it can actually get to the lock and then, be removed safely afterward. That's a good way to put it. So let's say we've got a drug and it's designed to target a protein in the brain. Well, first, it has to be absorbed. It needs to get into the bloodstream.
Whether it's a pill or an injection or whatever.
Right. And then from there, it has to actually travel to the brain. That's the distribution part.
And not all drugs can get past the blood-brain barrier.
Exactly. It's like a protective shield that tries to keep harmful stuff out of the brain.
Like a VIP pass only certain molecules can get.
I like that analogy. So then we have metabolism, which is where the body starts to break down the drug. This often happens in the liver,
Okay.
and this can affect how long the drug stays active and what kinds of byproducts it produces.
So metabolism is kind of like a behind-the-scenes team, transforming the drug into all these different forms.
Yeah, that's a good way to think about it. And then at the very end, there's excretion. That's how the body gets rid of the drug, through urine or feces.
The cleanup crew making sure things don't get backed up.
Exactly. Each step in this whole process can affect how well the drug works and whether it's safe. Luckily, our computational methods are getting much better at predicting and optimizing these properties.
It sounds incredibly complicated, like a delicate dance between the drug and the body. So where do you see all of this going? What are you most excited about for the future of computational drug design?
One area that I think is really exciting is quantum computing.
Quantum computing. That sounds so futuristic.
It does, doesn't it? But it's becoming more and more real. Quantum computers work so differently from the computers we use every day. They can handle these super complex calculations that would take our normal computers, years or even centuries to solve.
Like having a super brain that can analyze things in ways we can't even imagine right now.
That's a good way to put it. And think about what that could mean for drug design.
Yeah.
We could simulate molecular interactions with a level of accuracy that is just mind-blowing.
We could target those disease pathways with incredible precision.
Exactly. And that's not all. Quantum computing could also help us discover completely new targets by analyzing those huge biological data sets we talked about earlier, like having a search engine, but for new therapies.
It sounds like it could unlock so many possibilities. But what about AI? Where does its role go from here?
AI is already having a huge impact, and I think that's only going to grow. We're probably going to see even more sophisticated AI models developed specifically for drug design. Imagine AI that can not only design a drug, but also predict all those ADME properties, its side effects, even how it interacts with other drugs.
It'd be like having a virtual pharmaceutical company all inside a computer.
Yeah, that's a great way to think about it.
Speaking of futuristic stuff, the sources also mention these organ-on-a-chip systems. What are those all about?
Oh, those are really cool. They're miniature devices that basically mimic the functions of human organs.
Wait, so tiny hearts and lungs and things all on a chip?
That's the idea. This technology allows us to see how drugs interact with human tissues, but in a very controlled environment.
And that's better than just using, cells in a dish.
Yeah, it's much more realistic. And of course, it's a lot less controversial than using animals for testing.
A win-win for science and ethics. So how could these organ-on-a-chip systems change things?
I think they could really revolutionize how we test the safety and effectiveness of drugs, and they could even help us develop personalized medicine.
Personalized medicine?
Yeah, imagine making an organ-on-a-chip system that's specifically made to match someone's unique genetic makeup. That would let us test how different drugs would affect that particular person and really personalize their treatment plan.
That would be incredible. It's like getting a custom-designed medical treatment.
It really is. And it's all driven by these advancements in computational drug design.
It sounds like we're on the verge of some major changes in how we think about medicine, with computers and AI becoming more and more important.
Definitely. It's an exciting time to be working in this field.
It's pretty amazing to think about how far we've come, from these really hands-on lab experiments to all these powerful virtual tools. It's almost like we've traded in our test tubes for computer chips.
Yeah, it really is a huge shift. But at the end of the day, the goal is still the same. We want to create safe and effective treatments that make people's lives better.
Absolutely. And speaking of that, these advancements that we've been talking about have the potential to change how we think about personalized medicine.
Definitely. Just imagine a future where we can tailor treatments to a person's unique genes, their lifestyle, even the specifics of their disease, like a custom-made suit, but for medicine.
So instead of a one-size-fits-all approach, we're moving toward treatments that are as unique as the people getting them.
That's the goal. And computational methods are really driving that shift. By analyzing all that data and simulating these complex biological processes, we're starting to get a much deeper understanding of how different factors can affect how someone responds to a drug.
It's like having this super detailed map of each person's biology to guide us to the most effective treatment.
That's a great analogy. And as these tools keep getting better, we can expect to see even more personalized and precise therapies in the future.
It's a pretty incredible time to be alive,
Yeah.
especially if you're interested in this intersection of science, technology, and medicine.
Couldn't agree more. It's exciting to be working in a field where we can see these breakthroughs happening and contribute to developing treatments that could change people's lives.
Well, I think that about wraps up our deep dive into the world of computational drug design and virtual screening. It's been a fascinating look at how computers are revolutionizing how we find and develop new medications.
It's been great talking with you about it.
And as always, thanks to all of you for joining us on the deep dive. We'll see you next time.